this section, we are going to be looking at the test for starch in various food samples and the diverse procedures we can follow depending on the food sample. The test for starch is based on the formation of a colored complex with iodine. The iodine solution used is a yellow-brown solution of iodine dissolved in potassium iodide. When iodine solution is mixed with starch, iodine molecules fit into the helical structure of amylose, one of the types of starch, the other is amylopectin. This interaction produces a blue-black color. A negative test, that is the absence of starch, is observed when the food is stained with the yellow-brown color of iodine solution. A positive test, of course, that is the presence of starch, is seen by a blue-black color. When given a stock solution S, the test is straightforward. Put 2 milliliters of the stock solution S into a test tube. Using a dropper, add 3 drops of iodine solution to the test tube while shaking the mix. Then observe. A positive test will give a blue-black coloration, while a negative test will give a yellow-brown color. A sample report of the procedure is: 2 milliliters of solution S were added to a test tube using a syringe. 3 drops of iodine solution were added to the solution using a dropper. The solution was shaken to mix and observed. In other cases, we may be provided with solid foodstuff, say a tuber, for example, potato, a corm, for example, cocoyam, a grain of maize, fruits, for example, banana and apple, etc., to test for the presence of starch in them. We will not proceed with the formation of an extract as in previous tests. Here, we apply iodine solution directly to the foodstuff. Cut the potato, banana, and apple into 2 halves. Apply iodine solution to the cut surface using a dropper and observe. The iodine solution will turn blue-black upon contact with the potato tuber and cocoyam, while for fruits, apple, and ripe banana, the cut surfaces are stained yellow-brown. If we used unripe banana instead, there will be the formation of a blue-black color. A sample report of the procedure is: the potato tuber, banana, apple, etc., was cut into halves using a knife. Iodine solution was applied to the cut surface using a dropper and observed. For a maize grain, we will be required to evaluate the distribution of starch in the grain. To do this, we need a soaked maize grain to make it soft and easy to cut, and we will be required to make either of 2 cuts. A cut through the median plane to give 2 halves, or a cut by the side. The cut grain is then dipped in iodine solution and removed, then observed. We will observe that the embryo is stained yellow-brown. The cotyledon too will be stained yellow-brown, while the endosperm, the food store of the seed, will stain blue-black, indicating the presence of starch. For the observation and results, the apple and ripe banana stain yellow-brown, indicating the absence of starch. Solution S, potato tuber, and unripe banana turn blue-black, indicating the presence of starch. For the maize grain, the embryo and cotyledon were stained yellow-brown, indicating absent starch. The endosperm, the food store, turned blue-black, indicating the presence of starch. In conclusion, we say, no starch is present in ripe banana and apple, while starch is present in the potato tuber and unripe banana. We may be required to explain why unripe banana tests positive for starch and not banana. The reason is that during ripening, starch in banana is converted to sugars, for example, sucrose, glucose, and fructose by amylases. For the maize grain, we can conclude that food is stored in the endosperm of the maize seed in the form of starch. We have come to the end of this presentation. Our next presentation will be on the test for proteins. Thanks for your time. Hope you enjoyed following up the presentation. Please subscribe to this channel, like, and share. By subscribing, you will be getting notifications when new presentations are available.